Very good day, students. Today we are taking up a very important topic as for our paper is concerned, and this is about the osmoregulation. So, students, before starting this topic, I want to give you some of the concepts which are very important in osmoregulation. So, students, first of all, we have to take that what is the phenomenon of osmoregulation and what is the meaning of this particular word, the osmoregulation. So here I have written two things. First is the ionic and second is the water. So osmoregulation is actually the balance or the regulation of the salts, which means the ionic and the water. So osmoregulation is the regulation of the salts and water inside the body of the animals. Now this is a very important, you can say, a phenomenon which is present in each and every animal. If you start with some protozoans even, they are also osmoregulating. So there is a sort of regulation inside their bodies. So they are regulating the amount of the water inside their bodies. Now, if we go further, we have porphyrins. They are again regulating the amount of the water, and it is being maintained by some special cells which are called as myocytes. So myocytes can regulate the amount of the water that is coming inside their bodies. And this extracellular water current is maintained by the choanocytes. So as we have gone through these animals in our BSc. So we know that how they are regulating their water and ionic balance. Now many studies they have been taken on the body fluid concentrations in different animals, or you can say organisms, and majority of the marine animals, they have their internal osmotic pressure that agrees very closely with the seawater. So that is very clear that they are conforming with the environment in which they are living. Now suppose there is a marine invertebrate. It can be any marine invertebrate which is living inside the high salt concentration medium. So this particular animal, it will be conforming to the external salt concentrations, and the bodily concentration of the salt of this animal will be matching with the external environments. So this is a kind of, you can say, conformation in the homeostatic mechanisms of ionic and water regulation inside these animals. Now, if we go for some freshwater animals also, and these freshwater animals, they are regulating their body salts against the low concentration of the salts in the external environments. That means if one organism is living inside, suppose, a low salt concentration medium, it is maintaining a high salt concentration in its body fluids. So that is a regulator kind of a thing. So if we compare these two conditions, the first was the marine animal, which was conforming its body fluids with the external fluids. Second condition is of a freshwater animal, which is maintaining the salt concentration in its body fluids while living in the low salt concentrations. So taking these two conditions into account, we can go for a particular belief that the life has originated in the sea, because the high salt concentration which is maintained in the freshwater life is coming from the marine sea. So some of the scientists, they believe that the life has originated in the marine seas and then it has migrated to the freshwater life. So students, the marine invertebrates, uh, they are isoosmotic with the sea. So as the concentration of the water in the sea increases or decreases, they maintain an ionic composition of their body fluids, which is very comparable to the external environments. Now, this is again very common feature of the sealant traits also. So if we go with the sealant traits, the mesoglia of the Aurelia, it has got more potassium and very less of the sulfate. If you take the example of Aurelia, the potassium level in the plasma, if we take the concentration of this particular uh, element, the potassium, it is 106 as compared to the sulfates, which are only 47. So there is a huge difference between the concentrations of the so there is a huge difference between the concentrations of the potassium and the sulfates in case of Aurelia. Now, if we take some other example, say of one annelid, if we take the example of Arenicola, so here you have high amounts of sodium, potassium, calcium, magnesium, and even chloride in the plasma of these animals. And if we take some arthropods, suppose you take Garcinus as an example, so we have got again high concentrations of potassium, but here the magnesium levels are kept low at 34. If we take one example of mollusca, if we take sepia into account, we have got a high concentration of potassium. It can like move after, yeah, it can be above 200, and the level of the sulfate, it is quite low, and it is kept near to 20 or 25. So what I mean to say is that with these specific concentrations of the salts in the plasma of these animals, we are coming to a point that these animals, they can regulate their salt concentrations in the body fluids in the conformation to the external environments. Now students, if we go with the some examples of higher animals than these, if we take some examples of echinoderms, suppose, so they are having the high potassium values. And if we go with some polychaetes, they may show a reduced sulfate content, as we have seen in the Aurelia. So students, there are some like peculiarities of these body fluids, and now they are regarded as being the adaptive significances. So this is a kind of adaptability of these animals, that they can retain a low sulfate levels in their body fluids, and they can go for the higher concentrations of potassium, sodium, and calcium in their bodily fluids. So over the time, or you can say over the evolution, they have given rise to this kind of the concentration of the body fluids, and this is ionic regulation. Actually, this is regulation, or you can say osmoregulation. Now if we go with the typical definition of the ionic regulation, this would be the maintenance in a body fluid of the concentrations of ions differing from those that would result from a passive equilibrium with the external medium. So this definition of ionic regulation can give you a simple idea that what is regulation. Suppose if an animal is present in a particular medium and it is conforming with the external environment, if the external environment is changing with the suppose low sodium content, the body fluid of the animal is also now at the low sodium concentration. So this is called as conformity. But if this animal can maintain a high sodium levels inside its body against low levels in the external medium, so this will be the ionic regulation. And this is what is osmoregulation is all about. Now, if we go for a overall idea of this particular thing, so the calcium and the potassium, they tend to be more concentrated in the extracellular bodily fluids. So these two things are more concentrated in the extracellular bodily fluids, whereas the magnesium and the sulfates, they are less concentrated. So overall, this is the case. Now, if we go further, the distribution of the ions which is responsible for the potential drop across the cell membrane, it is known as the resting potential, as we have already uh, gone through this particular thing in the, you can say, nervous control or the impulse transmission. So if you have more of the positive ions on the outer side and negatives on the inner side of a nerve, then that is a kind of polarized nerve. And whenever the positive ions, they are coming inside and the negatives are going outside, that is called as a depolarization. Now this kind of depolarization, or you can say the formation of the action potential, it is again due to the uptake and the detake of the ions out of the nerve. So these action potentials, they are also the result of a kind of osmoregulation. Now, these ions are also required for the formation of this particular action potential, and these ions should be present in the body at a particular threshold level, or you can say at a particular concentration. If the animal is unable to achieve that particular threshold level of a particular ion inside its body, so there may be some hindrance in the production of the action potentials. So these ions, they are very, very important, and their threshold concentration, I must say, threshold concentration is very, very important of each and every salt in every animal. Now, some marine mammals, they are having some excretory organs too. So these excretory organs, what they are doing is that they are going for continuous outflow of the water, and they are taking out the excretion also. So that's why we take osmoregulation and the excretion as a combined, you can say, phenomenon. So they are both taken together because the removal of the water is very, you can say, an essential part of the excretion. So if you have to throw some nitrogenous waste products outside the body, you need water. If you have ammonia, you should have lots of water. If you have urea, it will take less of water. And if you have uric acid or the crystals or guanine crystal or something else, then you require less of the water. But water is very, very important to throw these excretory products out of the body. So thus, by controlling the removal of these ions from the body, the excretory organs, they contribute to the regulation of the composition of the body fluids. So as we have seen in our kidney, that our nephrons or kidney can throw some particular ions out of the body in the urine if they are more in the bodily fluids. And if these ions, they are necessary for the body and they are going out in the urine, those will be reabsorbed. So this is the function of the excretory organs, that they have to regulate the balance of both the water and the ions. So coming to the next slide. Now what are the osmoregulatory challenges? So first is the osmotic regulation, second is the ionic regulation, and third is the nitrogen excretion. So as we have discussed previously, that the control of the tissue osmotic pressure is again a very, very important thing for an animal. And this is a particular challenge of osmoregulation, that you have to control the tissue osmotic pressure, and you have to control the concentration of the ions in the body fluids uh, by not changing the tissue osmotic pressure. So you should maintain this osmotic pressure, as well as you have to excrete the things out, and you have to maintain a particular water concentration, or you can say amount of the water inside the body. So this is the first challenge. Now the second challenge is the ionic regulation. So here, as we have discussed earlier, that many animals, many invertebrates, they have to have some concentration of sodium, potassium, calcium, magnesium, chloride, sulfates, and so on for their bodily mechanisms. So they should have at least a threshold level of each and every salt in their bodily fluids. And next is the nitrogen excretion, as we have discussed just now, that the excretion process and the osmoregulation process, they are going side by side, and they are integral part of each other. So the pathway by which the animals excrete ammonia, it is again osmoregulatory in nature. As ammonia, it needs more of water uh, to get removed. Now, if we go with the consequences, now what are the consequences of this osmoregulation? So these can be the changes in the concentration of the ions. They have the potential to affect the structure and function of macromolecules. Now, as we have discussed, that the changes in the concentration of ions, they can affect the body mechanisms, as we have discussed the action potentials. So there should be threshold levels of each and every ion that is required for each, you can say, mechanism or the physiological process that is going inside the body of the animals. Now the second is the cells, they are exposed to osmotic gradients, can shrink or swell. Now this is again a very, very important thing, that these cells, as if suppose an animal is living in some freshwater environment, and it needs salts in the body fluids. So what it will be doing, that it will be excreting dilute urine, and it will be retaining the salts inside the body. And this particular thing will affect the cells of that animal, as we have discussed in the osmosis in the previous classes, that what is endosmosis, it will be the entry of the water into the cell. The cells will swell, and if there is exosmosis, the cells will shrink. So that's how this can be one of the consequence of the osmoregulation. Now next thing is that there is there may be some change in the cell volume that can damage cells directly. So suppose there is some endosmosis inside the cell. So the cell is swelling. So there is a change in the cell volume. So that can damage the cell. So cell can burst, or it can shrink to such extent that it will be damaged. Now students, these are some of the things which should be avoided by the animal. So if there is some high change in the concentration of the ions, if the cells are swelling or they are shrinking too much, or there is a large change in the cell volume, so the animal should avoid these kind of things. Now for that, the animals, they may have different mechanisms inside their bodies, that an efficient osmoregulation can take place. Now here, some of the things that an animal can have, those can be, suppose the first can be the presence of the excretory organs. So there can be some typical excretory organs that can excrete some water, and they can retain some salts, or they can do vice versa for the animal without changing the osmotic gradients of the cells of that particular animal. So that will be a very good, you can say, asset for that animal. So they can have the excretory organs. Secondly, these animals can have the reduced permeability. This is a very, very important second point, that whole of the animal can have a reduced permeability to water. Suppose there is excess of water that is coming inside the body of the animal. Now what the animal will do, that it will be excreting, or it will be throwing this water out, as the amoeba do, as the protozoans they do. So what they are doing, they are making contractile vacuoles, and they are throwing out the water which is coming inside their bodies. Now if the animal do something with the permeability of the outer membrane, that the water which was coming inside the body, now it is not coming because of the reduced permeability. So the animal can change some aspects of the osmoregulation. So that's why I take this particular point, the reduced permeability, as a very, very important point in osmoregulatory processes. Now students, the third thing which the animal can do is the improved active uptake of the salts. So this is a very, very important point again. Now this point means that a particular animal which is living in fresh water, suppose, so it is low in concentration of the salts, and the animal needs more of the salts in the freshwater environment. And what the animal will do, that it can actively uptake those salts inside its body. So that is a mechanism which is controlling the cell volume. It is also controlling the shrinkage or the swelling. It is also controlling the concentration of the ions in the body. So these three things can help the animal to reduce the amounts of these consequences in a particular animal. So after this, if we go for suppose the mechanism of this particular osmoregulatory consequence, then what is the mechanism? So we have got three types of solutions, as we have discussed in the previous classes. The first is the isotonic solution, which is very it is having the same concentrations of the salts inside the cell as well as the outside of the cell. So there is same concentration. That's why it is isoosmotic. Iso means the same, and osmotic refers to the osmosity. The second type is the hypotonic solution. Hypotonic, hypo refers to less of the salts. So here we have very less concentration of salts on the outside, and we have high concentrations of the salts towards inside of the cell. The third type is almost the vice versa of the hypotonicity. And now here we have high concentrations outside, and we have low concentrations towards the inside of the cell. Now this is again one part in your syllabus. It is specially mentioned in your syllabus, the three types of the solutions. Now if we take the mechanism behind these solutions, what is this? It is the osmosis. Now here, if you see this particular tube A and tube B, now here you can find that we have got a permeable membrane which is present in the center, and we have got less concentration of the salts on this side. We have got high concentration on the left side. So what we have is, we have a low concentration of the solution on the right side. We have high concentrated solution on the left side. So what will happen, the water, as the osmosis is the movement of water, and the water, it will go from less concentrated solution towards the high concentrated solution. So this is the movement of the water, and this is osmosis. So what will happen? The level of the water, it will rise on the left side as compared to the right side. So here the water level is decreasing, and here the water level is increasing. So this is the movement of the water with the ions, or the ionic difference, you can say. I give you one example. Suppose a particular human being, he is taking more of the salts. If he will take more of the salts, he will be having high BP problem. So there will be a condition which is called as you can say tension, hypertension. So that is high BP. Now why there is high BP? That if the man is taking more of the salt, that will be absorbed from the intestine, it will go to the blood, and when this salt will reach the blood in high amounts, it will attract water. So more of the water will be going into the blood. So when more of the water is reaching the vessels, so what will happen? The volume of the blood in the vessels will increase. So when the volume of the blood increases, the blood pressure also increases. So this is the mechanism of the increase of the blood pressure with the high concentration of salts. So there is a one basic, you can say, mechanism, or you can say the basic phenomenon, then that where you have more of the salts, it will take more of the water. So this is the basic phenomenon. If you have more of the salts in the body, it will attract more water inside the body. If you have less of the salts, there will be incoming of less of water. So this is a basic concept of endosmosis or exosmosis. Now students, if we take this concept to a higher level, or you can say on the level of the cells. So here we have a particular diagram which is showing the process of the coming in of the water or going out of the water of a cell. So this diagram is also very prevalent in the BSCs. Now here what we have the isotonic conditions, we have the hypotonic conditions, and we have the hypertonic conditions. Now in isotonic conditions, what will happen? That the amount of the water which is going in the cell and which is going out of the cell, they are equal. So that means there is no net movement of the water into and out of the cell. So the cell will remain as such in this particular condition. So 0.9% solution, or the saline solution, is isoosmotic to the blood. So we take, suppose we take any cell out of the body of a dissected animal, and we take in 0.9% of NaCl. So that will be isoosmotic to that particular tissue. So that will not be damaged because of this particular endo or exosmosis. Now, if we take the hypotonic condition into account, what will happen? If it is having less concentration on outside, more concentration towards the inside, what will happen? The water will come inside the cell. So that will result in the bursting of the cell. Now here the cell is bursting on this side, and vice versa happens in the case of the hypertonic solution. So here the water is going out of the body of the cell, and this cell is shrinking, and this is exosmosis. So students, this particular diagram can give you the idea that how the salts can regulate the amount of the water that is coming inside and that is going outside. Now students, if we talk about, suppose some protozoans. Now here, as we are talking about the cells, Now we are taking some protozoans first of all into account before we start the actual osmoregulatory process in higher animals. So we are taking up the protozoa into account first of all. Now here we have the very common example of protozoans, that is the amoeba proteus. Now amoeba proteus is a protozoan. It is a single-celled animal, and this is, I should say, this is animalcule. This is a very small animal, and that is present in fresh water. That means this is the case which is in amoeba. So it is living in a hypotonic condition. Now what will happen, that if it has got more concentration towards the inside and less towards the outside, the water will come inside the amoeba. And this is happening 24 hours, 7 days. So this amoeba is having the problem of water, you can say, incoming. So every time the water is coming inside the body of the amoeba. So this is a very big problem. Now what the amoeba has done to this problem, that amoeba has developed some contractile vesicles. Now these small vesicles, they are formed by the pinching of the plasma membrane. Now these are very small structures, the contractile vesicles, and these vesicles, they join together to form a big contractile vacuole. Now this contractile vacuole will do the function of osmoregulation. Now how it is performing the osmoregulation will be taken on. This particular contractile vacuole is performing two types of movements. Number one, this is a systole. Systole is the contraction of the contractile vacuole. And the second part is the diastole. So diastole is the relaxation. Now this contractile vacuole has got some contractile myofibrils over it, which are contracting and putting the contractile vacuole into the systole phase. This particular contractile vacuole, which is present in the cytoplasm of amoeba, it is taking in the water. It is collecting the water. Now some scientists, they have seen a particular dark layer surrounding this particular contractile vacuole. So there is a dark layer around contractile vacuole. Now when they see this particular layer in scanning electron microscope, now they found that this dark layer is of mitochondria. Now this is a very important discovery for amoeba. So all the contractile vacuoles, they are surrounded by many mitochondria, and these mitochondria, they are giving energy to this contractile vacuole, not for the contraction. This is very important thing to note, that mitochondria, they are giving energy to the contractile vacuole, not for the contraction but for the relaxation phase. Actually, when this contractile vacuole is in the diastolic phase, when it is relaxing, the water is pumped into this contractile vacuole actively. So this active transport of the water inside the contractile vacuole is maintained by energy, and that is maintained by the mitochondria. So this is the function of the mitochondria. Now here I can say that the animals, they must maintain their nutritive and the excretory relationships. Now the main thing is the animal, or the amoeba here, I should say, the amount of the water which is coming inside the body of the amoeba, that should be equal to the water going out. So that's how the amoeba can survive. If that is more than the outgoing water, it will like just burst up, just like this diagram. So there can be some mechanism to go for their homeostasis, or the homeostatic condition. The first can be that they should have their own metabolism. That means the water which is coming inside should be regulated to some extent by some organs. Now, if we are talking about these protozoans, there are no particular organs like that. So there should be the presence of the food vacuoles or the contractile vacuoles that can take some of the water inside them. And the third can be the surface permeability. So this is again a very, very important thing as we have discussed earlier. So the surface permeability that must present a particularly acute problem to the animal. So the permeability should be of such a level that the animal is not in a consequence or a bad consequence of osmoregulation. So that's we have already discussed previously. So here in amoeba, we have got large number of contractile vacuoles. If more of the water is coming in, more of the contractile vacuoles can be formed. So there can be like 10, there can be 5 in number at a particular time. If we talk about the second animal, which is paramecium, the paramecium is having two contractile vacuoles. And here also we have got this particular condition, which is the hypotonic condition. And paramecium is also living in fresh waters. Now here we have some difference than the amoeba. We have got two contractile vacuoles and they are fixed in number. Their position is fixed and their outpour is also fixed. So they cannot wander here and there in the body of the paramecium as they can in the amoeba. In paramecium, the outpour of these contractile vacuoles, they are on the dorsal side of the paramecium. As the paramecium has got a dorsal and the ventral sides, so they are opening on the dorsal side. These contractile vacuoles in paramecium, they have got some radial canals, and these radial canals, they are radiating towards all the sides in paramecium. And these radial canals, they are functioning to gather the water towards the center of the contractile vacuole. So as we draw these vacuoles as star-shaped contractile vacuoles, so the arms of the star, they are actually the radial canals. So I'm not going more into this particular topic, but here the contractile vacuole is also showing these two stages of systole and diastole, but the difference is only in the number and the position. If we go further, if we go for suppose the euglena, this is another example. So here it is lying very close against the so-called the reservoir in amoeba, in euglena, sorry. So here what we have is we have got two flagella. So these flagella, when they move, they make a water current and they can take in water in the food vacuole. Uh, some groups of small vacuoles, they contribute to the main one. So as in the case of the amoeba, here we can have small contractile vesicles and these are making big contractile vacuoles. Now, there can be some fibers which are associated with these contractile vacuoles. And the fluid, it cannot enter by filtration. Now, this is a mechanism. And since the total volume within this particular cell membrane would be not changed, so therefore during the diastole there must be active secretion of the water through the vacuolar wall. So as regards the systole, this is to contractile properties of the wall of the contractile vacuole. So its thickness, it suggests that a layer of structural proteins might be present in it, and this contractile vacuole is contracting because of these particular myofibrils that are present over it. So that is the case of euglena. So you can give more examples from the book. And if we take some cases of poriferans, if we take example of suppose sponge, we have taken a sponge called Cycon in BSc. So there were choanocytes present in the radial canals, and these choanocytes were having flagella, and these flagella, when they move, they are making water current, and that water current is going out of the osculum of the sponge. So that was very clear in our BSc classes, so I'm not going much into this particular thing. If we take some sealant rates, they are again taking in water. If we take the example of Hydra or Obelia, they are again living in fresh water, and they are taking water along with the food through their polyps. So polyps are the eating zooids over here, and there is no obvious sign of the surface protection even. So they have an external membrane which is called as the perisarc in case of Abelia. If we take the case of Sertularia, or you can take the Plumularia, Bougainvillea, these all are having cenosarcs around them, and these are the protective layers. So from where the water will enter into the bodies of these animals? This is only with feeding. So this is very important thing to note, that with the water which is taken in by these coelantrates, uh, that is becoming a part of the gastrovascular cavity or the coelomic fluid. Now, these animals, they are keeping a particular concentration of ions in this coelomic fluid, which is very different from the outside water. So that can be considered as the transporting mechanism of ions and the food to the different parts of the body by the animal. So that was the case in the coelantrates. Now, if we go further, I am taking some examples of, suppose this is the regulation of the internal environments. So here what we are taking is, we are taking the freshwater environments. So in the freshwater, what we have low levels of ions. We have got very less concentration of the ions. They acquire the ions against the electrochemical gradients. So this is the point which I was talking about, that these animals, if they have the active uptake of the salts, so then they can live a better life. So this is acquiring the ions against the electrochemical gradients. Now next is dispose excess water against the osmotic gradients. So this is very obvious that if some animals are living in low levels of ions, they require ions inside their bodies. So to conserve the ions, they have to give off the excess of the water out of the body. So this is kind of regulating the internal environment if you are living in low levels of ions. Now this slide is showing the regulation of the internal environment in the marine kind of environments. Now there will be a total vice versa kind of condition in the marine environments. So here we have high levels of the ions, and if we take the most important ions, those will be the sodium, chloride, that will be potassium, some of the calcium. So these are some of the important ions that are very much required in accurate quantities by the animal. So they must be expelled out. So this is a very important thing. So the animal, if it has got you can say a reduced permeability of the membrane, so these ions will not come inside the animal. Now this animal has to expel out the extra ions, and it has to obtain the water against the osmotic gradients. So this is a total vice versa thing, as we have discussed in the freshwater environments. So these animals will give out a concentrated urine, they will conserve water, and they will lose maximum of the salts in the form of the concentrated urine. So they may have some excretory organs associated for the osmoregulation, which will be forming concentrated urine. So they may have large, you can say, the proximal and the distal convoluted tubules in their nephrons, if they have, then. So these are some of the things that these animals will do to remain in the marine environment. Now students, this particular slide is giving you a comparative analysis of the animals which are living in the fresh water versus which are living in the marine water. So here you can very well see that if it is living in fresh water, if we take the first figure into consideration, so what they are doing is they have more of water outside and they have more of the water inside because they are living in fresh water. They have to expel out the dilute urine. They have to excrete out excess of water because they are living in the fresh water. So what about the salts? If you are living in fresh water, you have low salt concentration outside. So that means you need salts. So for that need, we have to actively uptake those salts through the gill sites. So here we have the active uptake, we have the dilute urine in these kinds of animals. And on the vice versa, if we are in the marine kind of environment, there will be, suppose if you go for the urine, first of all, this will be a concentrated urine. So they will be taking and they will be throwing out more of the ions and they will be conserving the water. So ions are actively secreted out from the body because they are living in excess of salts. Now, these can give out ions through diffusion through the skin, as we have discussed in the earlier part of this lecture, that the reduced permeability or excessive permeability to ions can help these animals to live in marine or freshwater environments. So here, the seawater is taken in through the mouth along with the food, and those salts which are coming inside the body through the seawater, they are actively secreted out, or they may diffuse out of the gills. So this is one of the mechanism by which these animals can regulate the amount of the salts in their bodies.